we'd like to be able to sample these kinds of systems with plants. You know, like you're doing in lab, you want to be able to go out and grab some leaves and make some cool measurements of the ecosystem and how ecosystem processing is happening. There's one problem that we're going to talk about, and that's plants are jerks. I was complaining so much about plants being jerks that Liam made this for me. So here's the jerk, I'm a jerk plant. These are the other plants, and it's sort of awesome. So plants do some things that mess us up and be able to use them as perfect samplers for our soil system. Okay? The first of those is this issue of how open is that pool that we're taking the nitrogen into the plant and reducing it. So it's not that much different than photosynthesis. There's a substrate, you're taking up something, in this case nitrate, and then a couple of different enzymes are acting on it. Okay? In nitrogen's case, it's these two, so you have nitrate reductase and glutamine synthetase, and they have fractionation factors. They fractionate just like Rubisco does, at about 15 and 17 per mil. Okay? But Dave Evans, back in 2001, did a bunch of hydroponic studies, and what he showed was that when you have really low nitrate in the tanks, and these are all wheat cultivars, you don't see these expressed at all. But as you bring it up and bring it up and bring it up, and you get up here high at 12 micromolar nitrate in the tank, you start seeing these really large fractionations happening. Okay? So what's going on? You can probably guess, because you've basically seen this in another form with every lecture that you go to, is that you have, what is this? What would you call that? From Gabe's lecture, what would you call that? So this is basically a Rayleigh distillation. So it's this where you have some kind of fractionation that's occurring, but if you use up this entire pool, at the end you have the same isotopic ratio that you started with. Okay? So you're having fractionation happening all the time, but it's using up the entire substrate. Okay? So in a plant, what happens is there's significant differences between the 15N of the plant and the solution that it's drawing from if the rate of uptake is, so if what's available for it to uptake is much higher than its assimilation rate. So it's either using up all that pool, or it has the ability to make a choice enzymatically and is fractionating to a large degree. The good news for most terrestrial ecology is that most of our systems are down here. Okay? So when you're in the field, the supply of nitrate and ammonium to the root system is it's not very big. Okay? So you never have its supply rate being outstripped by the assimilation rate. So everything that's available is being assimilated. Where you need to worry about this is if you were doing a hydroponic study, for example, and you said, man, I get weird N15 numbers in my plants, it's probably because you're giving it a ton of available nitrate on the exterior of the root. Could also come up if you're working in an ag system, where agricultural systems tend to have much, much higher rates of available nitrogen. So those enzymes may start to fractionate. Okay? By and large, in sort of natural system ecology, most people either ignore this or don't even know it exists. Okay? There's another problem. This is Fiona Soper. She was an isotopeteer in 2011, and she got so excited that she's going to work on N fixation rates in Texas in grass savannas. And the first thing she did is she went out and said, okay, I'm going to sample all the tree species that are there, and we'll be able to tell which one's a fixer, which one is this, which one is that. And she comes back with these data. These are the two nitrogen fixing species in the system, and these are a bunch of non-nitrogen fixing species. We have the same, so her prediction was, well, down here, these guys all have the same available soil solution N15. Their leaves should all look the same. Up here, these are both nitrogen fixers. They should both be 0. And what did you get? Something totally different. Nitrogen fixers don't look like nitrogen fixers at all, and the other species in there aren't tracking, if they have the same solution N15, they're reporting something different. And she got fascinated by this, and if I have time at the end, I'll talk about how she worked all this out. But a large part of it, what's going on, is that there's within plant fractionation. And what that means is that somewhere through the assimilation, the metabolism, and transport of nitrogen between taking it up in the roots and using it for growth, there's a change in isotope ratio. And Robinson has probably done more of this than anybody in basically ag plants. But here, if you look at, this is a kind of a, anybody know what that is? Komatsuma? It's kind of a cabbagey leaf thing. And tomato, if they sample, they give it a nitrate source, then they look at the whole plant, the shoot, and the root. And in some cases, you see, well, okay, the whole plant and shoot are pretty good recorders of this, but the roots are all weird, so we're getting some kind of fractionation back to it. In tomato, here's your beginning nitrate source isotopic value, and if you were to look at the shoot, it's double, okay? So you're having fractionation events happening through the plant body. So there's a pretty large literature that's very confusing. Best papers are by a guy named Jonathan Comstock, who's actually Jim's PhD student, that looks at some of these mechanisms. But as an ecologist, you're often not interested in that. You want something you can measure out in the field. So the advice that I give you is if you want to use plants to say, my 3 sites, or my 3 elevations, or my 3 places are different, and I want to use plants to assess that, use the same species whenever you can. Because then you totally bypass most of this, because there's a lot of fidelity within a species on how those fractionations occur within a plant. So it's very important what you pick your phytometer to be. So if you're really lucky, just work in Hawaii, because we only have like 4 species of trees and they grow everywhere. And so you just use that one species all the way through and you get around this, okay? The other way you can do it is not just relying on the leaves, and you begin to assess the isotopic ratio of different pools in the plant. Upside is you get a tremendous amount of information. Downside is you're going to do a tremendous amount of work. And I'll go into that a little bit more at the end. All right. So right about now is when most people say, I'm going to work with carbon, oxygen, and hydrogen because this sucks. But don't, it's awesome. First of all, less stuff is figured out with nitrogen isotopes, so there's lots more work to do. And secondarily, it's just cool. Do you have a question? No. All right. And people in this course have not been put off. So Pam Templer, she was in the class in 1998, was not scared because she was so empowered by all these awesome bits of information. She went on to do really cool stuff with nitrogen isotopes in trees. And this was a study she did in 2007. And basically the question, what she was interested in, is in the Northeast, there's a syndrome that we think there's very different rates of a lot of these processes depending upon what the dominant tree species is in that area. So we see different rates of mineralization, different rates of nitrification, denitrification, depending upon what species are there. And what Pam wanted to know is, okay, I can go out and do all this work and measure the rates, those process rates in those ecosystems, but can I sample the trees and see those differences in the isotopes? Are the trees recording it? And so she looked at beech, hemlock, sugar maple, and red oak, and then different pools of N15 from wood, foliage, litter fall, surface litter, fine root, organic soil. And what she found was there's just no information in the wood in terms of the nitrogen cycling. Now, John will probably say that's because you can't actually measure nitrogen isotopes in wood, and that could be true. You're getting close in foliage, but you're not really comfortable with what it's doing. But then there's a really, really close relationship with fine roots and organic soil. So she found this very powerful sort of thing for northeastern forests, because she could go back and look at what was the correlation between the rates that we measure in the system and the isotope ratios of fine roots and organic soil. And she got these really nice relationships. Okay, so here is the fine root, delta 15N, the organic soil, delta 15N, and this is the mineralization rate, and this is the nitrification rate. So process things that were very interested in ecosystem ecology. And she got really nice correlations. And this allowed her to then do isotope sampling on broad spatial scales and make inferences about what are the rates of nitrification or mineralization. And even more powerfully, how do they change? So we could do this every 5 or 10 years and say, well, rates of mineralization are going up or down for some outside perturbation, be it temperature, CO2, or whatever it is. Okay? We're going to do another isotopology here in a second, but I wanted to show you this. This is another one of those, I love it when people tell me really simple things. instead of getting lost in huge amounts of detail. And this is just a cross plot of 15N and delta 13C of what you generally see on Earth. And you've really seen that there's this big difference between C3 plants and C4 plants. And then today I've talked a little bit about nitrogen fixing things. They come down around 0. But I wanted to point out that the whole world isn't covered by land. A lot of it is aquatic. And the production dynamics in those other systems are different. And so the base of those food chains are also different. So you have some power in differentially telling the difference between, is this organism in a trophic system that's derived from the ocean, or is it from a trophic system derived from the land, something in between, a mixture of those two. And I just want you to pay attention to the numbers, because you can see, you might find an organism that it's down here around a -5. And you go, what? Those people at that lab don't know what they're doing. But the potential is there that they're in some kind of marine trophic system where the base of that food chain is marine production rather than terrestrial production. Now, why do I tell you this? If you thought the last figure was bad, I just taught in the Gulf of Maine for 2 weeks to all the students who were going to start freshman year of undergraduate. So they're primarily 17. And they've sampled isotopes. I gave them to a boat going by. They FedExed them back to Cornell. They ran them. They got the data back, and I gave them extra credit if they plotted it up and said something intelligent about this experiment they were doing. And the experiment they were doing was looking at tide pools. And they looked at 3 tide pools. One was very high, one was intermediate, and one was lower. So you can think of this as, this is really connected to the ocean. It's only exposed during the tide for a very short period of time. These upper pools are basically only getting spray and splash, and they're collecting fresh water, and they're doing other things. And they sampled the entire community there and did species diversity and all of this stuff, but they also collected isotopes. And then they sent this to me. This is one pool. This is the upper pool. It's a cross plot of 15N, 13C. This is N15 and carbon of one animal, Littorina littoria, which is a predaceous snail, okay? And this particular student presented what its isotopic value was in the lower pool, the intermediate pool, and the upper pool, and then this was the whole community of all the organisms that they had in that pool. So 2 minutes, and then say something intelligent about this.
Which pool is the top?
This is this upper pool, okay? This one's tougher. I gave them extra credit, but I still don't know what in the world they did, but I have some ideas. 2 minutes, say something really, really smart and cool. All right, do we have anything smart to say? No?
I have no idea if this makes any sense.
All right, Kelsey's going to say something really smart.
Or really dumb.
There's no real dumb.
Well, we were trying to, I was trying to make it now like the upper pool might be like a more tightly cycled nitrogen, because like you're not getting, it's not mixing with the ocean as often, so you're not getting replenishing like the ocean levels of nitrogen, whereas the lower pool is getting like new nitrogen input every tidal cycle. So I don't know if that would make much, but a nitrogen cycle, you're just getting, basically going through the system more often, going through the cycle more often, getting enriched.
Sounds smart to me. John?
I'm similar on that note. The lower pool, if you're getting more marine influences, are you going to get more marine algae?
Mm-hmm. They all have algae in them.
Yeah.
But, I mean, there's definitely more.
Going to be more marine algae, which is going to be more.
So let's ask them, like what I would ask this student is, okay, is this snail species more predatory when it's in these upper tide pools, or is it less? Would you say, how many people would say yes, it is? Yeah. Okay, what would happen, do you think, when that paper went to Seth back here? So what else, other than a change in trophism or where that organism is? What else could drive this difference?
See how we have different carbon sources.
Well, it's especially, I mean, it's huge differences in both carbon and nitrogen. So we're looking at.
What did you think?
It's the baseline of the food web, would be my guess. These numbers for this upper pool are actually pretty crazy, kind of odd. But one interesting thing about this site is it's a nesting gull colony. And basically what I think happens at the upper tide pools is, oh, is that what you're going to say? I think this is basically a guanotrophic food web up here. So you're basically having so much input from the gulls that you're getting it. Anybody have anything crazy to say about this?
I don't know if anything crazy to say, but kind of like everyone in the upper pool is theoretically eating the same thing, but there's still such a high variability. Like what the heck's up with that?
So what could drive variability in the nitrogen axis?
Where they're getting their nitrogen.
So it's where you feed in the food web. This is kind of cart before the horse because you haven't really had much food web stuff, but it enriches in each trophic level, so predators or more. So one possible explanation is you're looking at a bunch of predators feeding at different places, and you've got herbivores down here. This axis is a little harder to explain. Like
a 30 per mil.
yeah, how do you get that?
That was the range on that graph that we showed us.
So you know how you have to acidify your soils? Well, what do snails and crabs and almost every intertidal organism have? A shell with carbonates. So when they were, I didn't tell them not to do this, but in upper pools, the snails are really small. And so what they did is they put the whole snail, when they loaded, in the lower one, the snails are really big. And they smashed them with a hammer and pulled out the snail itself. So here, I think what you're looking at is a carbonate content of body gradient.
But that would also suggest that the only ones that have really high nitrogen values are the ones that are small.
Which would work if it's guanotrophic. Yeah, they're cool data sets. The one trap I wouldn't fall into is saying, oh my God, I got these data back, I plotted them up, I talked to people, we're all confused about it. Because I would say in my career, about 50-50, I have data sets that are sitting in a drawer that I still have not figured out, and the other half that first seemed impossible at first have been the highest impact publications I've ever had. Because the first time you look at them, you're like, what is this? And by the time you get to the end, it turns into something really cool. Okay? All right.